Okay, and welcome to today's lesson. You should have just completed the Amoeba Sisters video over protein structure and folding, so I'm just going to do a quick review of what was talked about in there and the 4 different types of structures that you can have for proteins. And then after completing this very short PowerPoint, you will be completing the worksheet Ribosomes and Protein Synthesis that begins on page 366 in your text. So protein structure and folding. As mentioned by the Amoeba Sisters in the video, there are 4 different types of protein structures that result from different types of folding, and we're just going to go through those. The first type that we are going to mention here is the primary structure. The primary structure is what produces the polypeptide chain. And that simply is each of the amino acids being covalently bonded in a nice straight line side by side. Okay. Now, sometimes, as Hank talked about in the video at the beginning of transcription and translation, we get a secondary structure, and this is caused because hydrogen bonds between amino acids not beside each other will create sheets and helices. Helices, just another word for spirals. What happens is, not only will they do like they do in their prime structure and hold covalent, but sometimes they will attach themselves every other, and this causes some to move forward and back, and then this creates what's called a pleated sheet. And in some cases, we will see spirals get created. Okay, that is the secondary structure of a protein. Okay, many proteins will have R groups attached to them. When those R groups start bonding, this forms a third structure called a tertiary structure. In this particular case, we start taking a more three-dimensional shape, and we get a lot of different bending and folding going on. Okay, so primary, secondary, and tertiary. This can happen to individual proteins. When we get to the fourth type called quaternary structure actually occurs when different proteins start bonding themselves together. So we'll have multiple proteins acting as one, and they fold around each other. The best example that I can give you of this is what happened. This is the hemoglobin in your blood. The hemoglobin in your blood is actually made up of multiple proteins working together. So again, you have the 4 different types of protein structure and folding that results. Again, if they're in a nice straight line where each one is holding on to the one beside it, we have the primary structure. In the secondary structure, amino acids will start bonding with those other than the ones that are right beside them. In a tertiary structure, the R groups that are attached to the amino acids start interacting not only with the ones right beside them, but the ones on down the line. And finally, if we start combining multiple proteins that function as one, we get a quaternary structure. Now, as I said earlier, you're going to move on to your worksheet, Ribosomes and Protein Synthesis, and again, that begins on page 366 in your book. Have a good day. Make sure you get your assignment completed and turned in before our next class.